Percentage of pollen germination. Aim: to calculate percentage of pollen germination. Materials required: 30 mg calcium nitrate, 10 mg boric acid, 10 g sucrose, 100 ml distilled water, needle, glass rod, dropper, spatula, hibiscus flower, compound microscope, cover slips, glass slides, blotting paper. Procedure: prepare the pollen germination medium by dissolving 10 g sucrose, and 10 mg boric acid, mg calcium nitrate into 100 ml of distilled water. Using a glass rod, stir the solution to mix it well. Using a dropper, take some nutrient solution and put 1 to 2 drops on a clean glass slide. Take the flower and sprinkle some mature pollen grains on the drop of medium using needle. Take a cover slip and place it gently on a glass slide with the aid of needle. Blotting paper, remove the excess medium from the slide. Place the glass slide under the compound microscope and observe the pollen germination. Observation: pollen grains start to germinate. After 10 minutes, observe the slide under microscope. We can observe different stages of germinating pollens. Some pollens are in the early stages of germination, while others have a long pollen tube. They consist of a protective coat containing several cells which are exine and intine. Also, the cell of the pollen grain divides into two cells. One of these is smaller in size and is called a generative cell, and the bigger one is called a tube nucleus. The intine grows out through the germ pore and forms pollen tube. By this time, we can observe the tube nucleus divides to form two male gametes. Calculation: to calculate the percentage of pollen germination, the number of pollen grains in a field of microscope is taken as N, and the number of germinated pollen grains in a field of microscope is taken as P. To determine the percentage of pollen germination, it is done using the formula P by N into 100. In the current microscopic field, here 2 pollen grains are germinated from a total of 5. Tabulate your observations and calculate the percentage of pollen germination. Repeat the process in 3 to 5 different microscopic fields. Conclusion: here we took hibiscus flower for the experiment. Although pollen grains of many species can be germinated in a nutrient-rich medium, but the percentage of germinations and the time taken for germination varies in different species. You can look for other species too.